Hello everyone. So we were doing catalysis, homogeneous catalysis, then after that heterogeneous catalysis. Then we did the theory of heterogeneous catalysis, absorption theory of heterogeneous catalysis. Now the next topic is enzymes. So enzymes are the biological catalysts, biochemical catalysts. These exist in the bodies of living beings, and basically these are the nitrogenous compounds, nitrogenous molecules, uh, and their function is same to enhance the rate of reaction inside the body or the biological reactions. Okay, so these are the nitrogenous molecules. Basically, these are the proteins of high molecular mass, right? Now, next is steps to enzyme catalysis. Now, there are two steps through which they functions. First is they bind to the substrate. Now, the substrate is the reactant molecule. So in biology, the reactants are known as substrate. So they first of all they bind to the substrate, then they form activated complex with the substrate. Their mode of action is again same as to the catalyst. They decrease the energy of they decrease the activation energy. They bring out the chemical changes at lower threshold energy value. They form the activated complex at lower threshold energy value with low activation energy, and hence they enhance the rate of reaction, right? So first they bind to the substrate molecule, then after that they decompose the activated complex and leave the product, and enzymes are then again regenerated for uh, reuse. So one mechanism, one famous mechanism of enzymes actions is lock and key hypothesis. And in this lock and key hypothesis, uh, the enzymes have some sites, active sites on them. So active sites means functional groups are present there. Some functional groups are present there, and the complementary groups are present on the substrate. Only those substrate are able to attach on an enzyme who have the complementary sites over them to these active sites. So active sites are the function group, and the function group which can attach to a particular molecule, particular atom. If those atoms are present on the substrate, then those substrate can attach to this enzyme. So binding of enzyme and the substrate will form the enzyme substrate complex. So once the enzyme substrate complex is formed, it forms activated complex which forms product, enzyme product complex. It changes to from activated complex to enzyme product complex. Enzyme product complex, which ultimately releases products and enzymes. Right? So this is the basic simple lock and key hypothesis. So enzymes have the active sites, and only those substrates are allowed to attach to it which have a complementary site to these sites. They bind together as key is fitted into the lock. So they form enzyme substrate complex, which changes to enzyme substrate activated complex, converted to enzyme product complex, and ultimately products and enzyme. Enzyme is again ready, again ready for the reaction, or you can say it is regenerated, right? Okay. Now the next is features, features of enzyme catalysis. So enzymes are highly specific in nature. They are they are very efficient in nature. A single enzyme can catalyze a million of molecules in minutes, and uh, again ready for reuse. So again, like catalysts, they are not consumed in the reaction. They will be recovered in the end of the reaction as such. The second feature of the enzymes is they are highly specific in nature. It means one type of particular enzyme is only for a particular substrate. Like the urease can hydrolyze only urea. Maltase can act only on maltose to convert into glucose. They work at optimum temperature, like as they are present inside the living organisms. So they are working with more efficiency at body temperature, which is 37 degree Celsius. So they work at optimum temperature. They work at optimum pH value also. So pH value, like in our body, is 7.4. They work at more efficiently at pH 7.4. There are two more terms, coenzymes and activators. So coenzymes are vitamins and activators are metal ions. Coenzymes are vitamins and activators are metal ions. So metal ions, which act as activators, can stick to the surface of the enzyme, and coenzymes acts along with the enzymes. So both coenzymes and activator enhances the activity of the enzyme. Whatever is the function of that enzyme, coenzymes, which are vitamin in nature, they come along with the enzymes. They act as promoter. They act as promoter. Like in catalysts, we have promoters. So here we have coenzymes and activators. Coenzymes are vitamins. They are not protein molecules. They are vitamins. And activators are mineral ions. So metal ions. Sorry, metal ions. So sodium, potassium, copper, cobalt, iron, like right? So these are the two new things, new terms which now enzyme inhibition. Like the functions of catalyst is killed by the poisons. On the same basis, the function of enzyme is also inhibited by the poisons. So the substances which inhibit the function of enzymes are known as poisons. Right? They work at optimum pH, they work at optimum temperature, they are highly specific in nature. Only a type of enzyme act on a certain substrate. Not a single enzyme can act on all types of substrates. They are very efficient in their work. Now the next is colloidal state. So colloidal state, so Thomas Graham was the person who first of all classified the substances on basis of rate of diffusion of their solutions. He took the solutions of the substances, then he studied the rate of diffusion of those substances through parchment membrane to animal membrane. So on basis of that, he divided the substances into two categories, colloids and crystalloids. Crystalloids and colloids. He categorized the substances which can pass through the parchment membrane very readily, very fast as crystalloids, like salt solution, like sugar solution, and those can diffuse very slowly through the parchment membrane as colloidal solutions, as colloids, as substance as colloids, and the solutions as colloidal solutions. Substances as crystalloids which can pass very fast through the parchment membrane, and solutions are true solutions. So silica, gum, like those are categorized under colloids. Okay. The next is now it has been discovered later on the crystalloid in one condition can be colloid in the other set of conditions, or vice versa also. Or vice versa also. Colloids can be crystalloids, or crystalloids can be colloids by changing the solvents, even solvents also, or by changing other conditions also, like pressure, temperature. Now here is the example. Sodium chloride behave as crystalloid in water. Sodium chloride behave as crystalloid in water. It forms true solution. It can pass through the parchment membrane very easily when it is dissolved in water. Whereas the same sodium chloride, if dissolved in benzene, it acts as chloride. It cannot pass through the parchment membrane. So later on, it was decided that it is the type of particles in the solution which decides the rate of diffusion of the solution of the substances, not the substances by their own, right? So one thing more is observed, that the substances having very large particles, and those large particles cannot move even slowly through the parchment membrane, they settle down, and those particles, those standing particles, are known as suspensions, right? So on basis of particle size limits and other distinctions, the solutions become of three types: true solution, colloidal solutions, and suspensions. Now the particle size, if less than 1 nanometer, they are termed as true solutions. If it is in between 1 nanometer to 1000 nanometer, then colloidal solutions. More than 1000 nanometer, then suspensions. True solutions are homogeneous, but that means their concentration is same throughout the solution. Colloidal appears as homo, but actually they are heterogeneous in nature. Suspensions are heterogeneous in nature. Now, simple example of these three is, for true solution, you can take the example of sugar in water, salt in water. So for this, sugar in water or salt in water. But whereas in this case, suspensions and colloidal, this can be understood by a single example or with the multiple examples also. For the single example, if we add some mud in the water and we observe that very large particles settle down. If we stir that mud continuously for 1 minute or 2 minutes, then we left it for some time, we'll see very large particles will settle down, and those large particles comes under the category of suspensions, whereas some particles will remain suspended. Some particles of mud, of clay, remain suspended in the solution. They will not going to settle down. Those form the colloidal solution. The simple examples of colloidal solution: Dettol in water, chalk in water, milk in water, right? Now, from where they can pass through the solution. So, in case of true solution, they cannot be filtered with the ultra filter paper or with normal filter. They can pass through even through the ultra filter paper. Whereas in case of colloidal solution, they cannot pass through animal membrane, but they can pass through the simple filter paper. But the suspensions cannot pass through filter paper or through the animal membrane also, and they can not pass through even through the filter paper. Their particle size is very large, right? Now, settling of set of particles on standings. So do not settle. They are very stable. True solutions. Again, the colloids are very stable in nature because they have Brownian moments in them. We'll study later on. Due to the imbalance forces in between their particles are always in motion, so they do not settle. Whereas in case of suspensions, the particles are very heavy; they settle with the time. True solutions, the particles are invisible. Here, in case of colloids, the particles themselves are not visible, but they can scatter the light passes through them, due to which the path of the light is visible, which is known as Tyndall effect. And dark particles can be observed under the ultra microscope, but not under the normal microscope. But whereas in case of suspensions, the particles are visible even with the naked eye. Now, next is diffusion. So, very quickly, true solutions particles diffuses very quickly, slides slowly, and suspensions do not diffuse. And then one physical difference between the three solutions is, in case of true solutions, they are very clear and transparent. In case of colloids, they are transparent like milk, like Midol in water, milkiness is there. And in case of solutions, opaque. Now the next is dispersion phase and dispersion medium. In true solutions, there are two terms. The substance which is going to dissolve in the other is known as solute, and the substance in which the solute is dissolved is known as solvent. So solute and solvent are termed as dispersed phase and dispersion medium in case of colloids. In case of colloidal solutions, dispersed phase, which is same as to solute, and dispersion medium, which is suspension. But in case of true solutions, we use the word that solute is dissolved in the solvent. But here in this case, the dispersed phase particles distributed in the dispersion medium and they suspended in the dispersion medium, right? So the particles which distributed in the dispersion medium in form of colloidal particles are known as dispersed phase particle, and the medium which provide and the medium which is a mean for the suspension, which the substance which is a medium for the suspension of the particles, colloidal particle, is known as dispersion medium, right? So this is all for now. We'll do classification of colloids in the next session, right? So thanks for now. Bye.